In this lecture, we will do a basic overview of some principles of medical genetics, in particular looking at the chromosome structure, and outlining the principles of mitosis and meiosis. So let's take a look at a review of chromosome structure. When we think about eukaryotic chromosomes, there are some terms and concepts that we need to review in case it's been a minute since we thought about these terms. We want to make sure that we lay a firm foundation so that for the rest of the course we are clear when I say, for example, the difference between a sister chromatid and a homologous pair, that everybody is clear on what these various terms mean. So when we're thinking about eukaryotic chromosomes and their structure, obviously eukaryotes come both in unicellular and multicellular forms. They have membrane-bound organelles according to the classic definition of what a eukaryote is. And one of the things that human beings do is we organize our chromosomes into a nucleic acid protein complex that forms, when it's condensed, this classic X shape that you can see here on this picture. And that structure is highly regulated. The relaxing and condensation of this structure plays a critical role in gene regulation. And this mechanism is made possible by the association of the nucleic acids to various histone proteins. And we will get into much further detail on the regulation, the relaxing, the condensation of the structure, and the role that it plays in allowing the cell to make incredibly detailed decisions in terms of regulating the whole central dogma machinery. So when we think about this homologous pair, like I said, I want to make sure that we're getting our terms correct. Human beings, of course, are diploid, which means we have a set of genetic information that comes from mom. We have a set of genetic information that comes from dad. There are haploid organisms. Bacteria, for example, are haploid. They only have one set because they're not sexually reproducing. But when we think about eukaryotes, there are no living haploid human cells. We produce haploid gametes, but haploid gametes are not a separate organism because they do not contain the full set of genetic information. So one of the definitions of what a human being is from a cell biology perspective is a diploid eukaryotic mammalian cell. Here's a picture of a karyotype. I'm sure you've seen this image many, many times in your previous courses. The standard chromosome number for human beings is, of course, 23. 22 of those we refer to as autosomes because they do not contain the critical genes for sex determination, and we have a separate pair of sex chromosomes. We'll do a lecture later in the course where we dive into the details of sex determination, both in humans, and we'll look at some unusual, at least from our perspective, strategies of sex determination in other animals. So here we have a diagram showing the two copies of the chromosome. You can see here we have the male, or the one that we receive from our father, that would be the paternal chromosome, and then the maternal chromosome. Now, this pair, the maternal and paternal chromosome, are referred to as a homologous pair. And the reason that they're referred to as a homologous pair is because the information on each chromosome is the same in terms of type, though not the same in terms of nucleic acid sequence. So what you see here is that from our father, we have received the dominant copy of allele A, and from our mother, we have received the recessive copy of allele A. Now, this example suggests that hair color is what is encoded by allele A, and so in this scenario, we have received one version of allele A from our father and one version of our allele A from our mother. So the nucleic acid sequence, in terms of the ATGC sequence, should not be identical. Right? The only way that would be possible would be if our parents were identical twins, which of course is not possible. So they have the same content, they have the same type of content, but not the exact same sequence content. They both contain the allele that will determine hair color, but they don't contain the exact same sequence. Now, another thing that students sometimes struggle with is the counting of chromosomes as we go through mitosis and meiosis. So here's a hypothetical eukaryotic cell. In this species, we have 4 chromosomes, 4 chromosome types, and then a paternal and maternal version of each type. So 8 chromosomes in total, because this organism is diploid. Now, if we want to divide this cell mitotically, we need both daughter cells to have a maternal and paternal copy of every chromosome. So somehow we have to take 8 chromosomes, divide it in half, and then still end up with 2 cells that have 8. Now, when we do DNA synthesis, and we're going to have a whole lecture on DNA synthesis, but when we do it, we do not create new chromosomes. So obviously one way to do this would be to take the 8 chromosomes, produce 16, divide them in half, and end up with 2 cells, each with 8. But that's not what happens. Instead, what we do is we double the chromatids, but not the chromosomes themselves. So we're going to do DNA synthesis, but we're not going to duplicate the entire chromosome. So here you can see we have 8 chromosomes and 8 chromatids. You can count chromosomes by looking at the centromere. So of course the centromere is the central attachment that's going to attach to the microtubule spindle. And so if we count the centromeres, we would see that we end up with 8 centromeres. 8 chromosomes, 8 DNA molecules. 1, 2, 3, 4, 5, 6, 7, 8. 8 centromeres, therefore 8 chromosomes. 1, 2, 3, 4, 5, 6, 7, 8. 8 chromatids, therefore 8 DNA molecules. Now, when we do DNA replication, we double the number of DNA molecules, but we don't double the number of chromosomes. So seeing here, in our classic X structure that we associate with chromosomes, we see that we still have 8 chromosomes. Count the centromeres. 1, 2, 3, 4, 5, 6, 7, 8. But look, the number of chromatids has doubled. So we don't double the number of chromosomes, we double the number of chromatids. I know that may seem pedantic, that may seem like wordplay that doesn't matter, but it's important to remember that we never are at a time when we have twice as many chromosomes as we should. And this is going to play into a role when we talk about being, for example, 2N or N, which is terminology you probably remember from a previous genetics course. Each new copy of the original chromatid is referred to as a sister chromatid. Sometimes I'll use the term twin sister, because unlike the homologous pair, the twin sister should have not only the same general content, such as the gene for hair color, but the exact same sequence. So in this example, we have chromosome 1 from dad. That's been duplicated, so it has that X pattern that we associate with chromosomes. Here we have chromosome 1 from mom. Okay, so in this example, our dad on the paternal chromosome has given us the allele for brown eyes, and our mother has given us the allele for green eyes. So chromosome 1 from dad and chromosome 1 from mom form a homologous pair. They have the same type of content, but not the same sequence. Now, if we looked at the twin sister chromatids, this chromatid on the left from dad should have the allele for brown eyes, and so should this sister chromatid on the right. Not only should it have the same type of allele, in this case the allele for brown eyes, they should have the exact same nucleic acid sequence. Now, of course, we're aware that when you copy DNA, there are occasionally errors. And so it may not be true that every single nucleotide is exactly the same, but sort of ignoring the concept, of errors produced by synthesis, the goal is to have 2 sequences that are exactly the same. So this chromatid on the left for dad and this chromatid on the right for dad are twin sister chromatids. This chromosome from dad and this chromosome from mom form a homologous pair. So during division, we need every daughter cell in mitosis to get a complete set from both the paternal and maternal chromosomes. So we're starting out here with 8 chromosomes, 16 DNA molecules. We're going to divide that into 2 cells, each with 8 chromosomes. So this cell here, if we count our chromosomes, 1, 2, 3, 4, 5, 6, 7, 8. This cell, if we count our chromosomes, 1, 2, 3, 4, 5, 6, 7, 8. So I know this is weird to think about it. We started with a cell that had 8 chromosomes. We divided 8 in half, and by the miracle of semantics, we have ended up with 2 cells that both have 8 chromosomes. There is no intermediate step here where we go from 8 chromosomes to 16 chromosomes. We double the chromatids. We do not double the chromosomes. So that's something that sometimes gets lost, you know, when you're going over this in maybe freshman biology or sophomore genetics or whatever it may be. But we want to make sure that we're getting the concept as clearly as possible. Here's another diagram. I'll do this throughout the course. Sometimes I'll show multiple versions of the same diagram so that it will click in your mind. I don't know, perhaps the previous diagram makes more sense to you. This one may make more sense. So sometimes I'll try to show different versions of the same thing so that one of them hopefully will click. Here is an unduplicated chromosome in a single bar with 1 chromatid. Here is a duplicated chromosome. This is still 1 chromosome, even though it has 2 chromatids, 2 sister chromatids. So briefly, let us go over some concepts of the cell cycle, mitosis, and then meiosis. When we think about the life cycle of the cell is divided into 2 large phases. The first phase is interphase, and interphase is the extended period of time between cell divisions. This is the time when the cell is doing its job, whatever its job or function may be. So if it's an epithelial cell, its job is to hold on to other epithelial cells and prevent fluid from leaking out onto the ground and killing the patient with dehydration. If it's a B cell, then its job is to be activated by T cells and produce specific antibodies. Whatever the job of the cell is, it's going to do that in the period of its life that we refer to as interphase, the period in between mitotic divisions. Then you have the M phase, which is the period of mitotic division. Now, if you've taken a biology of cancer course, you'll talk a lot about the regulation of the various checkpoints. We'll talk about that briefly, but obviously for a multicellular eukaryote, such as human beings and mammals, we want to make sure that we have a very tight control over mitosis, because dysregulated mitosis is going to lead to cancer. Cancer, by definition, is unregulated cell growth. And so there are going to be several opportunities within the cycle of any given cell that we're going to watch very carefully to make sure that all of the materials, information, structures are correct so that we can proceed through mitosis. No doubt you have seen a diagram like this in many previous courses. This is a simplified version of the cell cycle periods. Here we have a new baby cell created by mitosis. It's in its G1 checkpoint, doing whatever the structure of that cell is, all of the activity of that cell, whatever its job is, it's doing. It may pass through this first checkpoint, the G1S checkpoint that we'll say more about here in a moment. And if it does, it will go around the cell cycle through S, G2, and then eventually make it to mitosis. This is a slightly more complex version of the same diagram, illustrating the various major events of the cell cycle. During G1, it's doing its function. it should, if conditions are permissive, transfer from G1 to S. If, for example, it's an epithelial cell, when we think about epithelial cells, for example, on the surface of your skin, we don't want them to divide whenever they have an opportunity. Epithelial cells exhibit various strategies for preventing unregulated growth, and one of them is what we call anchorage dependence and dosage dependence. So if the cell is touching all around its perimeter other cells, the fact that it's touching a cell in every direction is telling it it doesn't need to divide. If the cell is laying on top of a stratum, some sort of substrate, that is telling it it does not need to divide. And so that epithelial cell may switch from G1 into G0 and enter a non-dividing stage because we don't need it to divide. We don't need our skin to divide unless there's a cut or a wound. And then that cut will create an open space next to the cell, which will be one of the signals to say we need to divide and fill in this gap. But it may be some other type of cell. We might think, for example, about the cells in the bone marrow that are going to go through constant division to replenish the white blood cells and the red blood cells. So we should pass from G1 to S, from S to G2, from G2 to mitosis. This point right here, which in this diagram is referred to as the G1S checkpoint, is also known as the restriction point or the R point. And that is because the restriction point is the most important regulatory step of mitosis. If the cell passes the restriction point, it is extremely likely that it will go all the way around the cycle and mitotically divide, assuming some disaster doesn't happen. For example, deleting a chromosome, losing a whole chromosome. That would stop division, or at least it should. It is very likely if we pass the G1S, also known as the R point, we will go all the way around the cycle. Therefore, the R point is the most important in terms of preventing cancer, because passing the R point in an inappropriate time is going to increase the likelihood that that cell divides when it should not, and unregulated cell division, as we've said, is the very definition of cancer. So there's a lot of special protections and regulations of this particular point. So you have undoubtedly, in many, many courses, gone over the steps of mitosis. I'm not going to belabor the steps of mitosis because my expectation is that at this level of a course, you have a general idea of what mitosis is and the steps involved. I do want, however, to draw some important distinctions between mitosis and meiosis. This being a medical genetics course, we're more interested in transmission of genes from one generation to another. So we're more interested in meiosis. However, to truly understand the advantages of meiosis, the structure and pattern of meiosis, we do need to review a little bit about mitosis. One of my objectives is that you would understand the similarities and differences between mitosis and meiosis. So as we think about the steps of mitosis, here we have the cell in interphase. It is living its life, doing its job, whatever that job may be. Then we transition into prophase. We have our duplicated chromosomes. They begin to condense. Remember that the chromosome's natural state is not this classic X shape that we think about in the karyotype. The DNA is very loosely organized in the nucleus. It has to be relaxed so that transcription factors can come and bind to it, activate gene expression. So it's not normally in this highly condensed structure. But now that we need to divide the cell, we're going to have to pack that chromosome from its very loose euchromatin state into its highly condensed heterochromatin state. And so you can see here in this diagram, here are our 4 representative chromosomes, one homologous pair, here's homologous pair 1, and then homologous pair 2. The nuclear envelope, of course, will begin to weaken because it will soon collapse so that we can move our chromosomes around. Here you see the mitotic spindle being developed, so we can do that. So as we move out of prophase into prometaphase, the envelope has collapsed, the mitotic spindle is assembling itself, and we're going to line up the chromosomes on the metaphase plate. Now, when we come here to metaphase, I really want you to look carefully at this arrangement. One of the things that I would really like for you to get out of this first introduction review is an idea of what mitosis metaphase looks like. So you see there's this imaginary equator right here in the midline of the cell that we refer to as the metaphase plate. And notice how the chromosomes are arranged. They're arranged in single file all the way down the line. They are not arranged in homologous pairs. For example, this chromosome's partner is all the way here on the bottom. So they're not arranged by pairs. They're sort of jumbled up, but they're in a straight line. Notice that the two legs, so to speak, of the chromatids straddle that line. And that's because when we pull on the kinetochore at the centromere, we're going to break these chromosomes like a wishbone. One chromatid will go to this cell, one chromatid will go to this cell. And that happens here in anaphase, right? So we've split these chromosomes in half, one section going left, one section going right. Now I want you to remember what this looks like, because when we talk about meiosis 1 and meiosis 2, how the chromosomes move when they are diploid, when they are haploid, is a function of the way they organize themselves in the metaphase plate. So now we have split our chromatids, our sister chromatids, and we're going to go through anaphase and then telophase. We will reestablish the nucleosome, the nuclear membrane, and we will have our two diploid daughter cells. Let's take a step back and consider the consequences and the goals of mitosis. This will be important for us to be able to understand the difference between mitosis and meiosis. So our goal here is to produce two cells that are genetically identical to each other and with the cell that gave rise to them. So the two daughter cells should be essentially clones of the mother cell. Now as I previously mentioned, there will be DNA synthesis mistakes, there will be small errors in that process, but the goal is for a perfect replication. Perfect replication never happens, but that's the goal. Every new daughter cell should have the full complement of chromosomes. So if the parent cell is diploid, the daughter cells should be diploid. And you'll also get roughly half of all of the organelle content. So another thing that we don't often think about in the cell cycle is when we're getting ready to do mitosis, we have to double the number of ribosomes. We have to double the number of mitochondria and lysosomes. We have to double the endoplasmic reticulum and the Golgi apparatus. You've got to get all of that content synthesized and doubled so that when the cell divides, everybody has enough organelle content, cytoplasm, etc., to be a functional cell. The key thing that we want to think about with mitosis, though, is that there should be no opportunity for diversity or variety in the daughter cells. And if we have a daughter cell that has variety or diversity, that means something has gone seriously wrong. An analogy that I sometimes think about is, if you've ever seen a Disney movie, almost all Disney movies have a similar setup in the beginning. And the setup is, there's someone, some young person, and they're part of a family or a community, and the way that that family or community thinks is backward and locked to the past. So the brave hero or heroine, has a dream that they want to be different, right? They want to go off and do something completely different than what they've experienced before. If you think about the original Beauty and the Beast, right? That's Belle's opening song. She says, I live here in this provincial little town where everybody is backward and everybody is small-minded, but I want to go out and I want to have a bigger life experience. That is not the attitude of the new baby daughter cells. We don't want any daughter cell to suddenly burst into some Disney song and begin to sing about the wonderful future that they want to be. If the parent cell is a epithelial cell, then the daughter cells must be clones of that epithelial cell. And if one of those cells suddenly decides to reactivate a large number of genes unrelated to being an epithelial cell, let's say, for example, genes related to being a hepatocyte, a liver cell, right? Insulin receptor, all of the mechanisms for producing and regulating gluconeogenesis and insulin, for example. Doing that, reactivating parts of the chromosome that cell type should not have, greatly increases the likelihood of cancer. And so there is no dream. There is no wonderful future. There is no rewriting the script for the daughter cells. They are locked into their system, and they are supposed to remain exactly as they are. And the reason that is that if they start to reactivate portions of their chromosome that they shouldn't have, the likelihood that they become cancer cells, they are transformed into cancer cells, is much higher. So there's a strict regulation. There's no dream for these daughter cells. There's no opportunity of a great new future in a worldwide somewhere. All of that is dead. They have to be exactly what we intend them to be. That's the goal of mitosis. No freedom, no diversity, no variety. You must be a cookie cutter of the previous cells. Now, in contrast, we're going to talk a little bit about meiosis. We'll go through the steps of meiosis. We'll go through the goals of meiosis. Now, some terms, just to make sure that we get these straight. Meiosis is the production of haploid gametes in a diploid organism. So we are a diploid organism. We will produce haploid gametes. Fertilization is the fusion of those haploid gametes to create a diploid organism. And the goal and consequence of meiosis is in complete contrast to mitosis. We want to maximize genetic variation in a safe environment. We don't want to promote cancer development. We don't want to promote dysregulated cell growth. But we want to maximize as much genetic variation as possible. So when we're thinking about the steps of meiosis, meiosis is broken down into two divisions. There's still the interphase where DNA synthesis and chromosomes are going to be replicated. There's the first division that we refer to as meiosis 1 and the second division, that's meiosis 2. The events of meiosis 1 and meiosis 2 are similar but not the same. In meiosis 1, as you can see here, we are going to deal with the separation of homologous chromosome pairs. And because we are separating homologous pairs, that means we are reducing the number of chromosomes by half. In meiosis 2, we will not separate the homologous pairs because we did that in step 1. We will instead separate the sister chromatids. So as we look at this diagram, we get an idea of these two different divisions that I was discussing earlier. So here in this situation, we have a cell that is 2N. We have a homologous chromosome pair 1 and a homologous chromosome pair 2. They're going to divide into two cells, A and B. Now notice that though there are two homologous pairs, there are 4 individual chromosomes. So when this cell divides into daughter cell A and B, where we had a total of 4, now we have 2 in cell A and 2 in cell B. Also note that it's the homologous pairs that have separated. So if we say, for example, that red is maternal and that blue is paternal, then cell A has received the maternal chromosome 1 and cell B has received the paternal chromosome 1. Cell A has received the paternal chromosome 2 and the maternal chromosome 2 is in cell B. So we went from 4 to 2. So we refer to that as the reductional division because the number of chromosomes have been reduced. Now in meiosis 2, we do not separate homologous pairs because we already did that. Instead, we separate sister chromatids. So in this scenario, the chromosome 1 that was maternal is divided into two different chromatids in cells A1 and A2, and then the same thing down here, B1 B2. So if we think about what's going on, first of all, we notice that in cell A1, none of the father's original chromosome 1 content is in that cell. It's all maternal for chromosome number 1. The opposite is true for chromosome 2. It's all paternal. Okay? Now what this means is that already we're getting some variation. The other thing that I want you to notice is that we started with 2 chromosomes, and what is our chromosome count after meiosis 2? It is still 2 chromosomes. So we start with 2 and we end with 2, which is why we refer to this as the equational division. So this is that same funny math that we observe in mitosis, where we started with 8 chromosomes, divided it in half, and ended with 8 chromosomes. So you need to be thinking, reductional division is meiosis 1. The reason it's reductional is because we divide our homologous pairs. The equational division is meiosis 2. The reason that it is equational is because we are not dividing chromosomes or homologous pairs, we're dividing chromatids. So the chromosome number stays the same. Another important thing we need to think about in meiosis is a special event that happens in prophase 1 of meiosis 1. So this event is not thought to happen commonly in mitosis. Now, this is an advanced genetics course, and so we can be a little bit more truthful maybe than we would be in an undergraduate course. I'm sure you've noticed this pattern, that things are more complicated than they're initially presented. So, I've taught freshman biology, I've taught sophomore genetics, and there's a lot of hand waving that goes on. So I'll be honest with you that the research indicates that crossing over does occasionally happen in mitosis. But the classical understanding, the textbook understanding of this event is that this event is for meiosis only. If it does occur in mitosis, the consequences are far less important because they only affect one small subpopulation of cells, whereas when it occurs in meiosis, it affects the entire new organism. Crossing over is one of the most important concepts that we'll discuss in this course, and it will come up again and again. So this is really something you need to wrap your head around because it's a topic we will be returning to multiple times. So it occurs in prophase 1, and what it is essentially the cutting and pasting of a portion of the paternal chromosome to the maternal chromosome. And this happens because a synapse forms between the homologous pairs, creating this tetrad in which the crossing over is going to occur. So what we see here is that a segment of the sister chromatid from one chromosome will cross over, sort of cut and paste, to the other sister chromatid. And this will create a brand new pattern of alleles that did not previously exist. So this occurs in prophase 1. Prophase 1 means prophase of meiosis 1. And it occurs through a series of processes. So there's several steps to prophase 1 we haven't discussed before. The leptotene step, the zygotene, the pachytene, the diplotene, and then diakinesis occurs. And what you'll see here is that the paternal and maternal chromosomes are relaxed. They are interwoven with one another. They begin to condense in preparation for meiosis. And as they do that, a synaptonemal complex will occur. Now, a synapse, of course, is a gap between any two structures. So a synaptonemal complex is a complex that is formed inside that gap that will draw these two sister chromatids of the homologous pair together. It will bring them into close association with one another. And what you'll notice is that one chromatid will cross over and lay on top of another. forming a cross-like structure we refer to as the chiasmata. When this happens, there will be a cut and paste event where you will get a new pattern that is brand new. Does not exist in the person creating these gametes, did not exist in their parents, will only be found in their gametes and potentially their offspring. In metaphase 1, we also need to pay attention to something unique. Metaphase 1 is where we're going to get the random alignment of homologous pairs along the metaphase plate. If you remember, I asked you to draw particular close attention to the way that the chromosomes align themselves in meiosis on the metaphase plate. I'm sorry, mitosis. We're going to contrast that to what we see here in a moment. Now, meiosis 2 is relatively simple. There's not as many important events that are occurring. It's very similar to mitosis. So let's go through and draw out some important events. Here we are in prophase 1. We've got the condensation of the chromosomes. They begin to pair up in their homologous pair. Note how closely they are connected to one another. You're going to get the synaptonemal complex forming and the crossing over that happens when they lay over each other, forming that chiasmata, and you'll get a cut and paste event. Now, when they do that, they will then move on to metaphase, and notice how they line up on the metaphase plate. Now, if we were to think about what it looked like in mitosis, if this is my metaphase plate, in mitosis, we had a linear arrangement of chromosomes. So if you consider this linear arrangement straddling the equator of the metaphase plate, that means that we must be dividing chromatids, because the chromatids are what straddle the equator. If you look here at metaphase 1, they're not in line, they're in pairs, just like the animals going into Noah's Ark, right? 2 by 2 by 2. And so if you look at where the alignment of the metaphase plate is, it's clear that this chromosome is going to go this way, and this chromosome is going to go this way. So we're not going to separate chromatids, we're going to separate homologous pairs. That happens, of course, in anaphase, and what we notice is here on, let's say, cell A, we are getting primarily maternal genetic information, with the exception of that crossed over piece. And then here on what will eventually be cell B, we're getting primarily paternal, with the exception of that little piece that's been crossed over. Now these will condense, there will be a slight interphase period, and then we will move on into metaphase 2. Now once again, observe the way that the chromosomes are aligned on the metaphase plate. Once again, like mitosis, they are linear, not in pairs. So metaphase 2 is setting up a chromatid division in a similar way that metaphase of mitosis sets up a chromatid division. So you get that division, and you end up with 4 separate cells. Now what I want you to observe as we look at these 4 separate cells is the diversity, the genetic diversity that we have already seen. So let's call this cell A, B, C, and D. Now in cell A, we have mostly maternal with paternal segments. Cell B, all paternal. Cell C, all maternal. And cell D, mostly paternal with maternal segments. So if you'll remember the goal of meiosis being to maximize genetic diversity, this crossing over event has created 4 genetically distinct gametes. These could be future eggs, they could be future sperm, whatever they're going to be. We have 4 potential diverse gametes. Now we've only done this diagram looking at 2 homologous pairs, chromosome 1 and chromosome 2. There are times when the cell will divide homologous pairs, and sometimes it will divide chromatids. This leads us to the question, how does the cell know when it is appropriate to separate chromatids and when it is appropriate to separate homologous pairs? Now, it's difficult in biology to discuss concepts without the fiction that the cell is sentient, that the cell is making decisions. We're going to pretend, obviously, that the cell is trying to decide, at this event I want to separate homologous pairs, but at this event I want to separate chromatids. In reality, of course, the cell is not making decisions. It's all a function of chemistry and biochemistry as the molecules and the structures function together. So the way that this mechanism works is the regulation of a structure referred to as cohesion. Now, cohesin is a molecule, it's a structure whose job is to hold the sister chromatids together. So let's start by looking at mitosis. In mitosis, we want to separate sister chromatids. When these sister chromatids are together, the cohesin is produced in preparation for mitosis, and that cohesin is doing its job holding those sister chromatids together. Therefore, to separate them, we must degrade this cohesin so that when the kinetochore pulls on the chromatid by walking down the mitotic spindle, it's able to separate the chromatids successfully. So this breakdown of cohesin allows the chromatids to separate. That's very simple. Cohesin holds them together. When you want to separate them, you remove the cohesin, therefore you're able to separate them. But in meiosis, we have 2 divisions, and we need to make sure that we only separate sister chromatids at the appropriate time, which would be meiosis 2. So if you look down here at meiosis, notice that there's going to be tension pulling on these chromatids at the kinetochore. And if we don't find a way to keep those chromatids together, when we pull on this kinetochore, we could rip off this chromatid. And that's going to produce gametes that have the wrong number of chromosomes. Now, this will be a common theme in the course. The ability of organisms to tolerate genetic diversity is very wide. Some organisms, like plants, can tolerate an astonishing array of genetic diversity. Human beings can tolerate, I would say, a moderate amount of genetic diversity, but one of the things that we do not tolerate well is variations in chromosome number. Most of the time, if you get the wrong number of chromosomes, as a zygote, you are not viable. If you are viable, you are likely to have very severe disease. You can probably think of some of the chromosomal number variation diseases, Down syndrome being the most famous example. So we need to make sure as a species, every cell, every gamete, gets the appropriate number of chromosomes. So the way that we do this, in meiosis, not in mitosis, is by the action of another molecule, Shigoshin. Shigoshin is a Japanese word that means guardian spirit. And so what Shigoshin will do when the cohesin is degraded in metaphase 1 to anaphase 1 in that transition, most of the cohesin will degrade. You can see it here. But some of the cohesin will be protected by the presence of that Shigoshin, and it will not degrade. Therefore, when we pull on those kinetochores, or more accurately, when the kinetochores walk back towards the centrosome, the sister chromatids are kept in a tight arrangement. Therefore, what we will then do is degrade Shigoshin in between meiosis 1 and meiosis 2. Then when we go into metaphase 2 and anaphase 2, we're able to get to that unprotected cohesin. We're able to separate the sister chromatids. Let me talk very, very briefly about gametogenesis. There are other courses in which you can go into this in more detail, cell biology, MedPhys 2, for example. We're going to just briefly touch on this, and because we're more interested in the transmission of the genes than on the production of the gametes themselves, but we need to discuss it just very briefly. Spermatogenesis, of course, is the production of the male gamete in humans. Oogenesis, the production of the female. Just as a note, anytime you see the prefix OO, it's always referring to eggs of some sort. So let's start with the simpler structure, the simpler process, I should say, male gametogenesis, spermatogenesis. You start off with a spermatogonium, and that spermatogonium is 2N, meaning it has a diploid number of chromosomes. It will enter prophase 1 and become what we refer to as a primary spermatocyte. Once it has entered prophase 1, it will have the crossing over event that we've already discussed, and at the end of meiosis 1, it will create 2 secondary spermatocytes. And because meiosis 1 is the reductional division, now our number of chromosomes has been cut in half. We have two cells with half the number of chromosomes. Therefore, up here, we are diploid. Down here, we are now haploid. Now, haploid cells cannot mitotically divide. You cannot divide a haploid cell into 2 haploid cells. This is the reason, for example, the medical community requires sperm donors and egg donors, because you cannot take a sperm and put it into a cell culture flask and give it all of the nutrients it needs and get it to mitotically divide. It can't do that. It does not have the necessary information. It does not have enough chromosomes to do that. So now that we're haploid, we're no longer a living organism. We have half of the information to form said organism. So the secondary spermatocyte will then undergo meiosis 2, and we will get 4 spermatids. They are also 1N, and then through a process we will not discuss because this is covered in other courses, these spermatids will mature and produce the functional sperm. So for the male, 1 spermatogonium, that is diploid, produces 4 haploid cells. In the female, it's a little more complex. If I was to make a overgeneralization of the reproductive strategy of the human male versus the human female has a quantity over quality strategy. Many of the sperm are not functional. Many of the sperm are going to be defective in some way. And so the male strategy is just to produce millions of sperm so that a few of those will be able to fertilize. This is why in males, sperm count is such a topic of discussion in fertility clinics. In females, you don't really talk about that. You don't talk about egg count. You talk about egg release. You talk about egg viability. But it's not about producing and releasing as many eggs as possible, which is what the male strategy is. So we start with a diploid oogonium. It enters prophase 1. Crossing over occurs. We produce a primary oocyte. That primary oocyte undergoes its division, and we get a haploid oocyte, a secondary oocyte. Now notice that rather than getting 2 oocytes, we also have this polar body. Now this polar body contains the other chromosomes. It will go off and it will be degraded. Only one of the 2 cells will move on into meiosis 2. Once we enter meiosis 2, the secondary oocyte will divide again, and we'll see the same pattern. There will be 1 ovum, and there will be a second polar body that is degraded. So for females, starting with a single oogonium that is diploid, will produce one haploid ovum. And that's what I'm meaning about the quantity versus quality difference between males and females. Let's talk a little bit more about the consequences of meiosis as they affect genetic diversity. So if we think about meiosis, you have 4 daughter cells in the male and 4 daughter cells in the female, though only one will actually go on and become a functional ovum. I'm still saying 4 because when it comes to probability, the probability is the same in the polar bodies. And so you still get 4 different cells, though only one will mature into a functional ovum. If we think about these haploid gametes, they're all going to be 1/2 of the possible chromosome number, and they should all be genetically different from the parent cell, but also from each other. And that genetic difference, of course, is the goal of meiosis and what separates it from mitosis. So what are the consequences of meiosis on genetic diversity? Well, if we think about broad strategies of reproduction, we of course have the asexual reproducing organisms and the sexual reproducing organisms. There are pros and cons from an evolutionary standpoint to both strategies. The pro of being an asexual organism like bacteria is that it's much cheaper and it's much faster. So once again, it's a quantity over quality strategy. There are more bacterial cells on the planet than any other type of cell. So in terms of evolution, in which success is determined by natural fitness, putting more offspring into the generation than your competitor, bacteria are extremely successful. So asexual reproduction is not a bad strategy. It's very successful. The pro is that it's cheap and that it's fast, and you produce a massive amount of offspring. The downside is that all of your offspring are essentially clones of one another. So the real benefit of being a sexually reproducing species is that every child, every offspring in the next generation should be genetically distinct from the siblings as well as from the parents. Now, the original source of these differences are going to come through either mutations or through errors of DNA synthesis. And so the original starting point of genetic diversity are the synthesis mistakes or the mutations that occur as a result of chemical mutagens, UV mutagens, etc. We will discuss various types of mutagens and their effect on nucleic acids at the very end of the course. We then take those differences and shuffle them together through sexual reproduction. There are 3 major mechanisms that contribute to the genetic variation that we see: independent assortment of chromosomes, crossing over, and random fertilization. It is very important that you understand these 3 concepts. Let's start with the independent assortment of chromosomes. The way that chromosomes line up on the metaphase plate of metaphase 1 is random. So what that means is there's an equal chance that the maternal chromosome is on the left or on the right for every pair of chromosomes. So if we take that into account alone, that produces more than 8 million possible combinations of chromosomes. So if we ignored crossing over, if we ignored random fertilization, any one individual, of all of the gametes they could potentially produce, there would be more than 8 million possible patterns. This is a diagram trying to illustrate what this would look like. Here in possibility 1, as you can see, cell A has received only paternal chromosomes, and cell B has received only maternal. Therefore, their gametes, the final gametes, are 2 that are all paternal or 2 that are all maternal. but it's completely random. The decision for chromosome 1 and the decision for chromosome 2 are statistically separate events. So just as likely is the possibility number 2, where cell A gets paternal 1 and maternal 2, and cell B gets maternal 1 and paternal 2. And as you can see, the outcome at the end is very different. This obviously grows as the number of chromosomes increases. So this is the exact same diagram as the previous diagram, but instead of looking at 2 homologous pairs, we're looking at 3. And so you can see that when you go through this independent assortment of chromosomes, you're drastically increasing the number of unique gametes. So this is just 3 chromosomes. Remember, we have 23 pairs. That's where that 8 million different combinations comes from. And that's simply based on the fact that in meiosis 1, the assortment is independent of every one. And what we mean by independent is this decision from chromosome 1 is completely independent of this decision for chromosome 2 and completely independent from chromosome 3. They don't influence each other in any way. I need you to remember this because when we start talking about Mendel and why Mendel's laws work the way that they do, it will be because of this independent assortment. Now we've talked about crossing over. Crossing over is another major contributor to genetic diversity. And that's because we create recombinant chromosomes. Now when you think about this word recombinant, what it means is we've taken the sections of the chromosome, we've separated them, and we have recombined them into a pattern that did not previously exist. In average, there is 1 to 3 crossing over events per chromosome pair. So once again, 23 different possibilities, and each one of those having 1 to 3. So this drastically increases the genetic diversity. So this diagram, I think, illustrates this nicely. In this example, my maternal chromosome 1 is dominant for A and dominant for B. Now this individual, right, if we think about this individual right here, this individual is heterozygous for A and heterozygous for B. They got their dominant alleles from their mom, they got their recessive alleles from their father. So the maternal chromosome contains all the dominant alleles. Paternal chromosome 1 is recessive for A and recessive for B. Now when crossing over occurs, and we're going to talk about this in a lot more detail for weeks and weeks, so I'm just laying the groundwork for future discussions. When crossing over occurs, notice that this chromatid, which originally had a dominant A allele, now has the paternal recessive A allele attached to the dominant B. That pattern did not exist until this event happened. So this is not something that the person got from their parents. If you'll remember, what they got from their parents was this over here. This is a completely new pattern. None of the cells in this person's body have a chromosome that is dominant for B and recessive for A. It does not exist in their cells anywhere. So not only is it unique to their parents, it's unique to their own autosomes. This is only in this one particular gametic tissue. And so the result is, when we look at the final outcome of this one division, is that we have one cell that has the unchanged maternal chromatid. We have one cell that has the unchanged paternal chromatid. And then we have 2 cells in the center that are recombined. They have a pattern that did not exist before. Once again, this can happen 1 to 3 times throughout all your 23 pairs. So that drastically increases the genetic diversity. Mitotic crossing over does occur, as I said earlier. It's something we associate with meiosis It does sometimes occur in a way that is clinically significant. It produces what we refer to as the familial form of retinoblastoma. And retinoblastoma may be a disease that we come back and discuss in more detail later. The third mechanism of genetic diversity is random fertilization. Now, when I say random fertilization, I do not have in mind the selection of a mate, because that process in humans is not random. You do not have access to every other member of the opposite sex of your species as a potential mate. Many of them live in other countries, they speak different languages, etc. And of course, our choice of a mate is not random. There are a variety of cultural mechanisms in which we choose who to mate with. So it's not statistically random. What we mean is that the union of all the possible sperm that the father could produce and the union of all the possible eggs that the mother could produce is random. And so when we think about adding up the effect of independent assortment, the effect of crossing over, the effect of random fertilization, there are about 70 trillion diploid combinations. Now, I'm a child of the 90s, and when I was a child, one of the big concerns, there's always fads related to the education of children, and the fad when I was a child was really big into self-esteem. So I remember being in kindergarten and having the little piece of paper, and you cut out the snowflake and you unfold it and you look around the room, everybody has created a different paper snowflake. And I remember my kindergartner teacher saying, every one of you is special. Every one of you is unique. You're your own little unique snowflake. And that's a self-esteem strategy, right? We want children to think, I'm special, I'm different, I have something to contribute. There's nothing wrong with that. It's actually way more true than my kindergarten teacher may have considered, because every individual organism of our species is a 1 out of 70 trillion different possibilities. So you really are a special, unique person. Your parents could have produced 70 trillion different offspring, and they picked you, right? And if that doesn't warm your heart and encourage you, I don't know what to tell you. Now, when we think about the evolutionary significance of this sexual reproduction strategy that prizes diversity really gives us a major benefit compared to asexual species. So natural selection is going to accumulate whatever genetic variations are favored by the environment. This makes us very resilient as a reproductive species. What this means is that we're all so genetically different, it is highly unlikely that any one or maybe even two brand new selective pressures could take us out as a species. I mean, consider the COVID-19 pandemic. Brand new virus that was not moving among our species before. There are very few people that have had any exposure to any virus similar to this. It erupts onto the scene worldwide, producing a very large amount of morbidity and mortality, and we have had no evolutionary exposure to this before. Nevertheless, there were people who were born in 1950, 60s, the 70s, the 80s, the 90s, the 2010s, the 2011, the 2000s, 2010s, and the 2020s, who had an inborn natural resistance to this virus, even though many of them were born before the virus even existed. And that is because we were all born with this very different genetic profile. You want your children to be genetically distinct from you as much as possible, so that if a brand new selective pressure enters into the world, like the COVID-19 pandemic did, or a future influenza pandemic, or any sort of stressor, natural selection stressor, that they will have the unique combinations that will increase their fitness. So that allows us as a species to be more stable. Now, that doesn't mean that some brand new stressor couldn't come in and wipe out 80%, 90%, 95% of the population. That's possible. It's also possible that the remaining 5% kill each other as they're fighting over food and resources and things like that. But a single stressor is unlikely to eliminate us as a species, or any sexually reproducing organism as a species. Species tend to go extinct if they're sexually reproducing when you have multiple stressors that stack up on one another. This is a major weakness for asexual organisms like bacteria. For example, if you think about when you have let's say you have an infection like strep throat. Strep throat is caused by Streptococcus pyogenes. And when you have an infection like this, you go to the doctor and they're going to give you a penicillin treatment, hopefully a penicillin shot, because that's a superior treatment. When they give you that penicillin shot, the thought process is that the bacteria that infected you has grown to a high concentration in your pharynx. And all of those bacteria should be genetically clones of one another. And therefore, if one of them is susceptible to penicillin, they should all be susceptible to penicillin. And so taking this drug should kill them all. So that means that the bacteria is extremely open to death through brand new selective pressures, such as the introduction of an antibiotic. Now, prokaryotes will attempt to make up for this weakness by using a process we refer to as horizontal gene transfer. Horizontal gene transfer is not something that we can do. We don't have the ability to do this. It's something that bacteria do quite frequently, and it is of extreme medical importance because it's a mechanism of increased antibiotic resistance and increased virulence factor transmission. So this is a diagram trying to illustrate horizontal gene transfer. Now, what I want you to pay attention to is that these arrows do not represent sexual reproduction. So this is not generation 1, generation 2, generation 3. Bacteria do not reproduce sexually. What's happening here is that this population, so we have the blue bacteria meets the red bacteria, and they share, red shares a portion of its chromosome with blue. But this is all within a single generation. Now, we do not have the ability to exchange genetic information with each other in a single generation. Human beings have a vertical genetic transfer. So I received my genes from my mother and father, and I passed on my genes and my wife's genes to my children. I cannot exchange genes with anyone within my generation. So for example, I can't go up to somebody who has red hair and say, wow, you know what, I really wish I had red hair. I've always wanted to have red hair. Could you give me the gene for red hair? And then we shake hands and all of a sudden my hair changes color to red. We can't do that. But that's exactly what bacteria will do. One bacteria will meet in the gut, like let's say a salmonella. It will meet in the gut and it'll look across at an E. coli, and it'll see that E. coli, and that E. coli will say, you know, I have a ciprofloxacin resistance. And the salmonella will say, I really wish I had ciprofloxacin resistance. Can you give it to me? And E. coli will say, sure, take this resistance gene. They're not mating with each other. They're not producing new offspring, little baby bacteria, but they're exchanging information within the same generation. And we don't have the ability to do that. That would be like I said, that would be like me going to someone with red hair and saying, giving me your red hair. But like the example that I just illustrated, it doesn't even have to be in the same genus and species, because salmonella is in one genus and E. coli is in another. So this is even across genus and species, or even higher orders of organization and taxonomy. So it would really be more like if I went to the zoo and I saw the zebra and I thought, you know, it'd be really nice to have black and white striped hair. And I walked over to the zebra and I rubbed my hand across its back on top of its fur, and the zebra gave to me the genes for striped black and white hair. And that is the sort of genetic shuffling that bacteria are able to do that we're obviously not able to do. This doesn't completely make up for the deficit in being an asexual reproducing organism, but it does make them much more versatile. And that's, once again, that's evidenced by the fact that we have major issues with antibiotic resistance and the production of new highly virulent strains. Okay, so that's the end of the first lecture. If you have questions, please feel free to reach out to me.